Every autumn, somewhere over the dark fields of northern Europe, a bird no heavier than a letter weighs the Earth's magnetic field against the stars and chooses a direction. It flies alone. No flock, no leader, no map. Just a small body hurtling through the night toward a destination it has never seen, guided by something inside its own eye. In 1972, researchers at the University of Frankfurt confirmed that European robins possess a magnetic compass, one that works only in the presence of light. That compass does not rely on iron. It does not rely on magnetism as we understand it. It relies on something far stranger, a quantum mechanical process unfolding inside a single protein molecule at body temperature in a living retina. The interaction it detects is over 1 million times weaker than the thermal energy surrounding it. Classical physics says it should be invisible, and yet the bird flies and it arrives. For more than 2 decades, no one could identify the molecule responsible. Then in the year 2000, a physicist named the protein, and in 2021, an international team proved it was magnetically sensitive. What they found inside the robin's eye may be the most sophisticated quantum technology in the natural world, and it has been operating for millions of years before humans ever split an atom. Today, we trace the discovery, decode the mechanism, and confront what it means for the boundary between physics and life. If this is your first time here, consider subscribing. And if you find this one rewarding, a like goes a long way. This is the quantum secret hidden inside a robin's eye. Let us find out what it knows. Every autumn, when the days shorten and the air cools across northern Europe, something stirs inside the European robin. It is not hunger. It is not fear. It is an urge, deep, ancient, and irresistible, to move. Ornithologists call it Zugunruhe, a German word meaning migratory restlessness, and it has been observed in caged songbirds for well over a century. A robin gripped by Zugunruhe does not sit quietly on its perch. It flutters, it hops, it throws itself against the walls of its enclosure in a particular direction, night after night, as though it can feel the pull of a destination it has never visited. What makes this behavior so remarkable is not the restlessness itself. It is the direction. Even sealed inside a windowless room, cut off from the stars, the sun, and every landmark on the horizon, the bird still orients, it still chooses, and it chooses correctly, aligning its body with the same heading it would follow if it were free, flying south across the continent toward the warmth of the Mediterranean. On the 12th of October, 1963, a young student named Wolfgang Wilczko stood in a magnetically shielded chamber at the University of Frankfurt and watched this happen for the first time under controlled conditions. He had placed European robins inside orientation cages fitted with small perches arranged in every compass direction, and he had recorded which perches the birds favoured. The room was sealed. No light from the sky could reach them. No sound from outside could guide them. And yet, when a magnetic field was present, even an artificial one generated by Helmholtz coils, the robins oriented in their expected migratory direction. When the field was removed, or weakened beyond a certain threshold, the preference vanished. The birds became random. Wilczko, working alongside his supervisor, Friedrich Merkel, published the results in 1965 and 1966. The claim was simple and extraordinary. European robins could detect the Earth's magnetic field and use it as a compass. The scientific community was not impressed. Prominent researchers, among them Erwin Stresemann in Germany and Donald Griffin in the United States, had already investigated whether birds might sense magnetic fields. They had found nothing, and they had concluded, publicly and firmly, that such a sensitivity could be ruled out. The idea that a small songbird might possess an entire sense that humans lack, one with no known receptor organ and no obvious physical basis, struck many as implausible at best. For years, Wiltschko's findings were treated with skepticism bordering on dismissal. Other laboratories tried and failed to replicate the results, often because they used different cage designs or magnetic field strengths outside the narrow range the birds could detect. It was not until 1972 that the tide began to turn. Wolfgang Wiltschko and his wife Roswitha, herself a trained biologist who had been studying sun compass orientation in homing pigeons, published a paper together in Science that changed the shape of the question entirely. Their experiment revealed something no one had expected. The robin's magnetic compass was not like a handheld compass. It did not distinguish between magnetic north and magnetic south. When the researchers reversed the direction of the magnetic field, the birds did not reverse their heading. They carried on exactly as before. What the robins were reading was not polarity, it was inclination, the angle at which the Earth's magnetic field lines dip toward the surface. Near the poles, the field lines plunge steeply into the ground. Near the equator, they run almost parallel to it. Everywhere in between, they slope at some intermediate angle, and that angle changes predictably with latitude. The Wiltschkos showed that the robin's compass distinguishes poleward from equatorward by reading which direction the field lines tilt, not which end of a magnet is which. This is called an inclination compass, and it was entirely new to science. At the time, the significance of this distinction was not fully appreciated. It would take decades before anyone understood why it mattered so deeply. But the inclination compass was a clue, a fingerprint of the underlying mechanism, and it pointed away from anything resembling a conventional magnetic detector. The breakthrough in acceptance came a few years later, not in Frankfurt but in Ithaca, New York. In 1974, the Wiltschkos travelled to Cornell University at the invitation of Bill Keaton, an entomologist who had independently discovered that homing pigeons use the Earth's magnetic field. There, working with ornithologist Stephen Emlen, they tested indigo buntings, a North American migratory songbird, in artificially altered magnetic fields. The study was rigorously double-blind. Wolfgang Wiltschko shipped his custom-built orientation cages across the Atlantic by boat. Neither the Wiltschkos nor Emlen had contact with the birds or the raw data until the experiment was complete. The results, published in Science in 1976, confirmed that indigo buntings orient magnetically. For the broader scientific community, this was the moment. The skepticism did not disappear overnight, but it lost its footing. Yet even as the existence of a magnetic compass became harder to deny, the mechanism behind it remained completely opaque. And the deeper researchers looked, the stranger the compass became. The most troubling clue arrived through light. Across a series of experiments spanning the 1980s and 90s, the Wiltschkos and their collaborators demonstrated that the robin's magnetic compass does not work in darkness. It requires light, and not just any light. Under wavelengths from ultraviolet through to green, the compass functions normally. Under red light, it fails. The birds become disoriented, as though the magnetic field has simply ceased to exist. In 1992, Wolfgang and Roswitha Wiltschko, working with Australian collaborators Ursula Munro and Hugh Ford, tested silver eyes, a small migratory bird native to Australia, under red, blue and green lights separately. The reasoning was elegant in its simplicity. If a photopigment in the eye is responsible for magnetoreception, then not every wavelength of light should be able to activate it. The silver eyes oriented normally under blue and green light. Under red light, they could not orient at all. This was a pivotal finding. It meant the magnetic compass was not merely located somewhere near the eye, it was photochemical. It required a specific kind of light to function, the same way a camera requires an exposure to capture an image. Something inside the bird's eye was absorbing photons of a particular energy and using that energy to detect the direction of the Earth's magnetic field. But what kind of molecule could possibly do that? The Earth's magnetic field is staggeringly weak, approximately 50 microtesla, roughly 100 times feebler than a common fridge magnet. The energy of its interaction with a single molecule is more than 1,000,000 times smaller than the thermal energy jostling every particle in a living cell. In the language of physics, this means the magnetic signal is completely buried in noise. It is not just quiet, it is quieter than quiet. Under every known principle of classical chemistry, no molecule should be able to detect it. And yet the robin detects it. Every autumn, every spring, across thousands of kilometers, in darkness, through cloud, over open ocean. The compass works. By the late 1990s, the question was no longer whether birds could sense the magnetic field. The question was how, and the answer, when it finally began to emerge, would come not from biology, but from quantum physics. The answer had in fact already been written down, 2 decades before anyone was ready to hear it. In the mid-1970s, a young physicist named Klaus Schulten was working at the Max Planck Institute in Göttingen, studying a class of chemical reactions that most scientists considered a curiosity. These reactions involved radical pairs, fleeting, unstable molecular states in which 2 molecules each carry an unpaired electron. Schulten had shown that the outcomes of certain radical pair reactions could be influenced by magnetic fields, even relatively weak ones. The effect was real, measurable, and grounded in quantum mechanics. But it was a laboratory phenomenon confined to test tubes and spectroscopy. No one had connected it to biology. Schulten made the connection. He had been following the work coming out of Frankfurt, the Wiltschko's demonstrations that the robin's compass was insensitive to field polarity but dependent on light. 2 features that, to a physicist, pointed in a very specific direction. A compass based on radical pairs would be inherently blind to polarity, because the quantum process that makes radical pairs magnetically sensitive does not distinguish between a field pointing one way and a field pointing the other. And it would require light, because radical pairs are born from photochemical reactions, reactions initiated by the absorption of a photon. In 1978, Schulten, together with Charles Swenberg and Albert Weller, published a short paper proposing that the avian magnetic compass was based on exactly this mechanism, a magnetically sensitive photochemical reaction with radical pairs as transient intermediates taking place somewhere inside the bird. The paper was 5 pages long. It appeared in Zeitschrift für Physikalische Chemie, a respected but specialist German journal. It was not Schulten's first choice. He had originally submitted the idea to Science. The rejection was swift and memorable. One reviewer wrote that a less bold scientist may have designated the idea to the waste paper basket. Schulten later recalled scratching his head and deciding it was either a great idea or an entirely stupid one. He chose great and published it where he could. Shortly after, he gave a lecture at Harvard, where he had been a graduate student. In the audience sat Dudley Hirschbach, a Nobel Prize-winning chemist, who listened carefully and then asked a question that would shape the next 20 years of Schulten's thinking. "In a test tube, you need light," Hirschbach said. "Where is the light in the bird?" Schulten knew immediately that this was the right question. If the mechanism was photochemical, then the compass had to be in a part of the body that receives light. The obvious answer was the eye. And if it was in the eye, then it was almost certainly entangled with the visual system, not a separate sense organ, but something woven into sight itself. The idea was elegant, it was physically sound, and it was almost entirely ignored. For 22 years, the radical pair hypothesis sat at the margins of both physics and biology, too speculative for experimentalists, too biological for physicists, and lacking the one thing that could make it real: a candidate molecule. Schulten had described the mechanism, he had described the requirements, but he could not point to a specific protein inside a bird's eye and say, "This is the one." The molecule, when it finally emerged, came from an unexpected direction. To understand what happens inside that molecule and why it matters requires stepping into a world that behaves nothing like the one we experience at human scale. A radical pair begins with light. A photon strikes a molecule and delivers a jolt of energy, enough to dislodge an electron from its usual position and send it hopping to a neighbouring molecule. What remains behind is a molecule missing one electron. What receives it is a molecule carrying one extra. Each of these molecules now has an unpaired electron, an electron without a partner, and that makes each of them a radical. Together they form a radical pair. What makes this pair special is not the chemistry, it is the quantum mechanics. The two unpaired electrons, having been born from the same event, are not independent. Their spins, an intrinsic quantum property, roughly analogous to a tiny internal rotation, are correlated. They exist in a shared quantum state, entangled in a way that has no counterpart in everyday experience. At the moment of their creation, the pair is in what physicists call a singlet state. The two spins are anti-parallel, pointing in opposite directions, perfectly balanced. But that balance does not last. Each electron sits inside a different molecule, surrounded by a different arrangement of atomic nuclei. Those nuclei are themselves tiny magnets, and they interact with the electron spins through a force called hyperfine coupling. Because the two electrons experience different hyperfine environments, they begin to precess, to wobble, at slightly different rates. This difference in precession drives the pair to oscillate between two quantum states: the singlet, where the spins are anti-parallel, and the triplet, where they are parallel. Back and forth, singlet to triplet, triplet to singlet, millions of times per second. This is the quantum dance, and it is exquisitely sensitive to the world outside. When an external magnetic field is present, even one as weak as the Earth's, it adds a small additional influence to the precession of each electron. The field nudges the spin dynamics, subtly shifting the rhythm of the singlet-triplet oscillation. Over the lifetime of the radical pair, this shift accumulates. It changes the proportion of time the pair spends in each state, and because the chemical fate of the pair depends on which state it occupies at the moment of recombination, singlet pairs can react, triplet pairs cannot. The magnetic field ends up altering the products of the reaction. Different orientations of the molecule relative to the field produce different chemical yields. The reaction becomes directional. It becomes, in effect, a compass. This is where the mechanism crosses from remarkable into something that borders on impossible. The energy of the Earth's magnetic field acting on a single electron is absurdly small. It is over 1,000,000 times weaker than kBt, the thermal energy that shakes every molecule in a living cell at body temperature. In classical chemistry, an influence this faint would be swamped instantly by random thermal motion. It would be like trying to hear a whisper in a hurricane. The signal should not survive. But it does survive because the radical pair is not a classical system. The singlet-triplet oscillation is a coherent quantum process, a delicate, synchronized dance between two entangled spins that exists outside the reach of thermal noise for just long enough to register the field's direction. The coherence does not need to last forever. It needs to last about 1 microsecond, 1/1,000,000th of a second. In that sliver of time, the quantum state of the radical pair accumulates enough directional information from the Earth's magnetic field to bias the chemistry. Then the pair recombines, the coherence is lost, and the result is locked in. 1 microsecond. That is the window. It sounds vanishingly brief, and by human standards it is. But for a quantum process operating at the molecular scale, it is an eternity. Long enough for the spins to complete thousands of oscillations, long enough for the magnetic field to leave its mark. Think of it this way. 2 coins are flipped at exactly the same moment, and as they spin through the air, an impossibly faint breeze, so gentle that no instrument could measure it, pushes against them. If the coins were heavy and spinning fast, the breeze would make no difference. They would land randomly. But these coins are not heavy. They are quantum coins, and they spin in a synchronized way that amplifies even the faintest push. By the time they land, the breeze has tipped the odds, not by much, not every time, but consistently enough across millions of radical pairs created every second across the surface of a retina to produce a signal, a pattern, a direction. That is the radical pair mechanism. It is a quantum process operating in warm, wet, chaotic biological tissue, extracting directional information from a force that classical physics says should be invisible. And in 1978, Klaus Schulten described it with remarkable accuracy, decades before anyone could prove it, and decades before anyone could name the molecule that makes it real. The molecule would not remain nameless for much longer. In the year 2000, 22 years after Schulten's proposal was dismissed and forgotten by most of the scientific world, a paper appeared in the Biophysical Journal that changed everything. Thorsten Ritz, working alongside Sali Adem and Klaus Schulten himself at the University of Illinois at Urbana-Champaign, did what no one had managed before. They named the molecule. It was called cryptochrome. Cryptochrome is a protein, a flavoprotein to be precise, found not just in birds but across the living world. Plants carry it, insects carry it, mammals carry it, humans carry it. It was first characterized in Arabidopsis thaliana, a small flowering plant beloved by geneticists, where it serves as a blue light photoreceptor involved in regulating circadian rhythms and controlling how seedlings grow toward the light. In animals, cryptochromes play a similar timekeeping role, helping to synchronize the body's internal clock with the cycle of day and night. None of this, on the surface, had anything to do with magnetism. But cryptochrome had a property that set it apart from every other photoreceptor known in vertebrates. Buried inside the protein is a small molecule called flavin adenine dinucleotide, FAD, a yellowish organic cofactor that absorbs blue light. When a photon in the right wavelength range, roughly 400 to 500 nanometers, strikes the FAD molecule, it excites an electron into a higher energy state. That electron does not simply fall back. Instead, it hops, transferring from the flavin to a nearby amino acid called tryptophan. But it does not stop there either. Inside cryptochrome, 4 tryptophan residues are arranged in a chain, each one positioned to receive the electron from the last, like stepping stones across a stream. The electron cascades down this tryptophan chain, moving further and further from its origin until it reaches the final residue. What remains is a radical pair. The flavin, now carrying 1 fewer electron, is a radical. The terminal tryptophan, now carrying one extra, is a radical. Their unpaired electrons are spin correlated, born from the same quantum event, entangled in a shared singlet state and sensitive to the direction of any external magnetic field. This was precisely the kind of system Schulten had described in 1978. A photochemical reaction, activated by light, producing a magnetically sensitive radical pair. And cryptochrome did not just satisfy the theoretical requirements in the abstract, it satisfied them in the eye. By the late 1990s, cryptochrome had been found in the retinas of several animal species, including birds. Ritz and Schulten saw the opportunity immediately. Their 2000 paper did more than name the molecule. It built a model. Because the eye is roughly spherical, cryptochrome molecules distributed across the retina would be oriented in every possible direction relative to the Earth's magnetic field. Each molecule's radical pair would experience the field from a slightly different angle, producing a slightly different chemical yield. Summed across the entire retina, these tiny differences would create a pattern, a modulation of the visual signal, a kind of filter laid over normal sight that shifts and changes as the bird turns its head. Ritz proposed that birds might not merely sense the magnetic field, they might see it. A shadow, perhaps. A wash of brightness and darkness, symmetric around the axis of the field lines, rotating smoothly as the bird changes direction. Not a sixth sense at all, but an extension of the one sense they rely on most, vision. The proposal was extraordinary, but it rested on a foundation that was, at the time, still largely theoretical. Cryptochrome formed radical pairs, that much was known. The radical pair mechanism could in principle detect weak magnetic fields, that much was established. But no one had yet shown that cryptochrome itself was magnetically sensitive. And no one had found it in the right place, in the right cells, in the right species. The paper was a map, not a destination. The journey to confirm it would take another 2 decades, and it would require an entirely different set of tools. The search began in the eyes of migratory songbirds. Henrik Moritzen arrived at the University of Oldenburg in northern Germany with a background in bird migration and a growing conviction that the answers lay at the intersection of biology, neuroscience, and physics. In the early 2000s, when the Ritz and Schulten paper was still fresh, Moritzen and his colleagues set out to answer the most basic question the hypothesis demanded. Is cryptochrome actually present in the retinas of migratory birds? And if so, where exactly does it sit? The answer came in 2004. Working with neurobiologist Reto Weiler, Moritzen's team demonstrated that cryptochrome protein and neuronal activity markers were present together, co-localized, in the retinas of European robins during magnetic orientation behavior. The same year, Andrea Müller and colleagues at the University of Frankfurt independently confirmed the finding in a different migratory species. Cryptochrome was there. It was in the eye. And it was active precisely when the birds were using their magnetic compass. But which cryptochrome? Birds, it turned out, carry not one but at least 6 different isoforms of the protein: Cry1a, Cry1b, Cry2a, Cry2b, Cry4a, and Cry4b, each encoded by different genes or produced by alternative splicing, and each occupying a different position within the retina. Cry1a was found in the outer segments of ultraviolet and violet-sensitive cone photoreceptors. Cry1b appeared in retinal ganglion cells, but only during the migratory season. Outside of migration, its expression dropped to background levels. The others were distributed in various patterns across the retinal architecture. The field narrowed its focus gradually, and the evidence began to converge on one candidate above the rest. Cryptochrome 4, specifically the isoform known as Cry4A, sat in a uniquely promising location. It was expressed in the outer segments of double cone and long wavelength single cone photoreceptor cells. These cells are not random structures. Their outer segments contain stacks of parallel membrane discs called lamellae, layered with extraordinary regularity, separated by roughly 1520 nanometers. This architecture matters profoundly because for a radical pair compass to work, the magnetoreceptor molecules must be anchored in place and at least partially aligned. A protein tumbling freely in solution would sample every orientation equally and the directional information would wash out. But a protein embedded in, or tethered to, an ordered stack of membranes would maintain its orientation, and the compass signal would survive. Cry4A had something else that set it apart. In European robins, its messenger RNA does not rise and fall with the 24-hour circadian cycle the way most clock-related proteins do. Instead, it is expressed at a roughly constant level throughout the day and night, exactly what you would expect from a sensor that needs to be permanently available, not one that switches on and off with the hours. More telling still, Cry4A expression increases during the migratory season. Researchers at Oldenburg compared robins with domestic chickens, a non-migratory species, and found no such seasonal increase in the chickens. The protein appeared to be tuned, in migratory birds, to be most abundant precisely when the compass is needed most. There was also a deeper evolutionary signal. Cry1 and Cry2 are extremely conserved across all bird lineages. Their sequences barely differ from species to species, suggesting a stable, ancient function, most likely related to the circadian clock. Cry4A, by contrast, shows strong positive selection and high variability between species. This is a hallmark of sensor proteins. Molecules under active evolutionary pressure to optimize their performance for the specific demands of each species' ecology. A clock gene looks the same everywhere. A sensor gene is constantly being refined. None of this was proof. Each piece was circumstantial. The right location, the right expression pattern, the right evolutionary signature. But taken together, the evidence formed a picture that was becoming difficult to dismiss. Of all the cryptochromes in the avian retina, Cry4A was the one that looked most like a magnetoreceptor. It was in the right cells, on the right membranes, at the right time, and evolving under the right pressures. The question was no longer whether the molecule existed. The question was whether it actually worked. While the search for cryptochrome was playing out at the molecular level, a parallel investigation was reaching into the brain itself. And what it found there would reshape the entire conversation about what it means for a bird to sense a magnetic field. In 2005, Henrik Moritzen teamed up with Eric Jarvis, a neuroscientist at Duke University, to look at something no one had examined before. Which regions of the avian brain become active when a migratory bird orients magnetically at night? Their tool was a protein called Zenk, an immediate early gene product that acts as a marker of recent neuronal activity. When a brain region fires intensely, Zenk expression rises. When it is quiet, Zenk stays low. By mapping Zenk expression across the brains of migratory songbirds tested during magnetic orientation, the team could ask a precise question: where in the brain does the magnetic compass lave? The answer was a region they named Cluster N. Cluster N is not a single structure. It is a collection of interconnected areas within a part of the bird's forebrain called the visual wallst, a region already known to process visual information arriving from the eyes. In night migratory European robins and garden warblers, Cluster N showed intense neuronal activation during nighttime magnetic orientation. The signal was strong and unmistakable. But when the same experiments were run on non-migratory species, zebra finches and canaries, tested under identical conditions, cluster N was silent. No activation at all. This was not a general arousal effect. It was not simply that migratory birds were more active at night. The activation was specific to a defined brain region, specific to migratory species, and specific to the conditions under which magnetic compass orientation occurs. When the bird's eyes were covered, the activation vanished. Whatever cluster N was responding to, it depended on visual input. 2 years later, in 2007, Dominic Hyers and colleagues at Oldenburg traced the wiring. Using neuronal tracing techniques combined with behavioral experiments, they demonstrated a direct anatomical pathway connecting the retina to cluster N through the thalamofugal visual system, the same pathway that carries ordinary visual information from the eyes to the higher processing centers of the brain. The magnetic signal, it appeared, was traveling along the same neural highway as everything else the bird sees. It was not arriving through a separate channel. It was embedded in the visual stream. Then came the decisive experiment. In 2009, Moritzon's team chemically inactivated cluster N in European robins, temporarily disabling the region while leaving the rest of the brain intact. The results were clean and striking. Birds with a silenced cluster N could no longer orient using their magnetic compass. They were lost. But their sun compass worked perfectly. Their star compass worked perfectly. Every other navigational system in the bird's brain continued to function as normal. Only the magnetic compass was gone. This was as close to a smoking gun as the field had produced. Cluster N was not merely correlated with magnetic orientation, it was necessary for it. Remove cluster N and you remove the magnetic compass, nothing more and nothing less. The implications ran deeper than anatomy. If magnetic information is processed within the visual system, carried by the same pathways, handled by the same brain regions, then magnetoreception is not a vague internal feeling, not some diffuse awareness of direction operating beneath conscious perception. It is processed as vision. The bird's brain treats the magnetic field the way it treats light and colour and motion, as something seen. Whether the bird experiences it as a pattern, a shadow, a wash of colour, or something entirely alien to human imagination, no one can say. But the neural architecture tells a clear story. The magnetic compass is a visual sense. Think about what that means. A robin standing in a garden, turning its head to scan for worms, is simultaneously looking at the world through a filter that no human has ever perceived. The sky, the soil, the hedgerow, and layered over all of it, something else. Something that shifts as the bird turns, something aligned with the invisible architecture of the planet's magnetic field. Two realities, superimposed, experienced as one. Moritzon himself, when asked what a bird might see, offered a careful answer. Perhaps it is a kind of shadow, he said, superimposed on whatever else the bird would normally be seeing. But what exactly the bird perceives, he added, we do not know, because we cannot ask the bird. The neural evidence was compelling. The molecular candidate was identified. But the field still lacked one critical piece: proof that the radical pair mechanism could actually operate at the strength of the Earth's magnetic field. In laboratory chemistry, radical pair reactions had been shown to respond to magnetic fields many times, but always to fields far stronger than the 50 microtesla of the geomagnetic field. No chemical system, natural or artificial, had ever demonstrated sensitivity at Earth field strength, let alone directional sensitivity. In 2008, a team at the University of Oxford set out to close that gap. The experiment was led by Kiminori Maeda and Christian Timmel, working with Peter Hore, a physical chemist who had spent years developing the theoretical framework for radical pair magnetoreception, alongside photochemist Devens Gust at Arizona State University. Their approach was deliberately indirect. Rather than testing cryptochrome itself, which was still fiendishly difficult to isolate and purify in sufficient quantities, they chose a synthetic molecule specifically designed to mimic the electron transfer photochemistry that cryptochrome performs. The molecule was a triad, 3 chemical units linked end to end. At one end sat a carotenoid, a long organic pigment of the kind found naturally in carrots and autumn leaves. In the middle sat a porphyrin, a ring-shaped molecule closely related to the heme group that carries oxygen in blood and the chlorophyll that captures light in plants. At the far end sat a fullerene, a hollow cage of 60 carbon atoms shaped like a football, the molecule sometimes called a buckyball. It was an extraordinary object, this triad. Nothing like it exists inside any living creature. But its physics, the way electrons move through it when light strikes, closely paralleled the photochemistry of cryptochrome. When green light hit the porphyrin, it excited an electron, which jumped from the carotenoid at one end to the fullerene at the other, a distance of roughly 4 nanometers. The result was a radical pair, the carotenoid carrying a positive charge, the fullerene carrying a negative charge, their unpaired electrons spin correlated and quantum entangled. And here was the breakthrough. The lifetime of that radical pair, the time it took before the electron jumped back and the system returned to its ground state, was measurably altered by the application of a magnetic field at or below 50 microtesla, Earth strength. The weakest field the researchers tested still produced a detectable effect. More importantly, the effect was not merely sensitive to the strength of the field. It was anisotropic. It depended on the direction of the field relative to the orientation of the molecule. This is the property that separates a mere magnetic sensor from a compass. A compass must not only detect that a field exists, it must tell you which way it points. The Oxford Triad did both. The paper appeared in Nature in 2008, and it landed with force. For the first time, a chemical reaction had been shown to function as a compass at geomagnetic field strength. The molecule was artificial. The experiment was conducted in frozen solution at low temperature, not inside a living cell. But the proof of principle was established. A radical pair created by light responding to the direction of a field no stronger than the Earth's. The physics worked. The question had shifted. It was no longer whether a chemical compass was possible. It was whether the one inside the robin's eye, built not from carotenoids and fullerenes, but from flavin and tryptophan, operating not in frozen solution, but in warm, wet, living tissue, could do the same thing. And answering that question would require something far more difficult than synthesizing a molecule in a laboratory. It would require extracting the bird's own protein, purifying it, and measuring its response to a magnetic field, a task that would consume the next 13 years. Before the protein could speak, the noise almost silenced it. In 2004, Thorsten Ritz, the same physicist who had named cryptochrome as the candidate molecule 4 years earlier, published a study that would become one of the most cited papers in the magnetoreception field. Working with Moritzen and the Wiltschkos at Frankfurt, Ritz asked a deceptively simple question. If the magnetic compass depends on a quantum process inside a radical pair, what happens when you flood the environment with oscillating electromagnetic fields at the right frequency to disrupt that process? The physics behind the question was precise. A radical pair in its singlet state oscillates between singlet and triplet configurations at a rate governed by the hyperfine coupling between its unpaired electron spins and the nuclear spins of nearby atoms. For a flavin-tryptophan radical pair of the kind found in cryptochrome, the relevant hyperfine frequencies fall in the low megahertz range, roughly 1 to 10 megahertz. An external oscillating field tuned to these frequencies would interfere with the delicate interconversion between singlet and triplet states, scrambling the quantum coherence that allows the compass to function. The static geomagnetic field nudges the spin dynamics in a consistent, directional way. A broadband oscillating field at the right frequency would overwhelm that nudge with noise. Ritz designed the experiment carefully. European robins were placed in orientation funnels under the natural geomagnetic field, with and without a weak broadband radio frequency field in the low megahertz range. The intensity of the added field was vanishingly small, far too weak to produce heating, far too weak to stimulate nerves, far too weak to be detected by any known biological mechanism other than a radical pair process. And yet the effect was immediate and total. Birds exposed to the oscillating field could not orient. Their magnetic compass was gone. Remove the field and the compass returned instantly. The disruption was clean, reversible, and frequency-specific. This was powerful evidence, not just because it showed the compass could be knocked out, but because of what it ruled in. The radio frequency effect was predicted specifically by the radical pair model. No other proposed mechanism for magnetoreception, not magnetite crystals, not electromagnetic induction, not any classical process, would be sensitive to such weak oscillating fields at those particular frequencies. The result was a diagnostic test, and the radical pair mechanism passed it. But it was what came next, a decade later, that turned a laboratory finding into an environmental crisis. In 2014, Moritzen led a study that appeared in Nature, and its findings were disturbing. For years, his team in Oldenburg had struggled with an inexplicable problem. Robins tested in wooden huts on the university campus could not orient magnetically. The same species, tested in the same kind of apparatus, had oriented flawlessly for the Wildschkoes in Frankfurt for decades. Something was different in Oldenburg, and no one could identify what. The breakthrough came when Moritzen's group lined the orientation huts with aluminium sheeting, effectively creating a Faraday cage that blocked external electromagnetic radiation. Inside the shielded huts, the robins oriented perfectly. Outside them, they were lost. The team then measured the ambient electromagnetic environment on campus and compared it with the levels inside the shielded huts. What they found was that the Oldenburg campus, like virtually every urban environment on Earth, was bathed in anthropogenic electromagnetic noise in the megahertz range. Radio broadcasts, electronics, wireless devices, power systems, all of them contribute to a low-level haze of oscillating electromagnetic fields that blankets modern human habitation. The levels were extraordinarily weak by engineering standards. They were far below every guideline set by the International Commission on Non-Ionizing Radiation Protection. They were thousands of times below the threshold considered biologically relevant for any conventional mechanism. But for a compass built on quantum coherence, a compass that detects the Earth's magnetic field through spin dynamics sensitive to oscillating fields in precisely this frequency range, the noise was devastating. The disruption was not caused by a single powerful source. It was the accumulated electromagnetic fingerprint of modern civilization, invisible and inescapable, interfering with a quantum sense that had evolved over millions of years in electromagnetic silence. The implications stretched far beyond a laboratory in northern Germany. If urban-level electromagnetic noise can disable the radical pair compass, then every migratory bird flying over or through a city, an industrial zone, a suburb, a motorway corridor, any environment saturated with anthropogenic radio frequency emissions, may be flying without its primary magnetic sense. The birds do not crash. They have redundant navigational systems, star compasses, sun compasses, landmark recognition. But the magnetic compass is the one that works on overcast nights beneath cloud cover, in conditions where the others fail. Losing it does not ground the bird. It makes the bird more vulnerable, more dependent on backup systems, more susceptible to disorientation at precisely the moments when the magnetic compass matters most. Moritzen was careful in his language. The study demonstrated that the effect was real, repeatable, and specific to the frequency band predicted by radical pair theory. It did not claim that all migratory birds everywhere were affected, but it raised a question that no one in the field had previously considered. Is the electromagnetic environment of the modern world incompatible with a sense that predates human civilization by tens of millions of years? That question remains open. What was not open, what could no longer be postponed, was the central challenge the entire field had been circling for 2 decades. Every piece of evidence pointed toward cryptochrome 4 as the magnetoreceptor. The neural pathway had been traced. The disruption experiments matched the radical pair predictions. The synthetic compass had proved the physics. But no one had yet shown that cryptochrome 4 from a migratory bird could actually respond to a magnetic field. In 2021, a paper appeared in Nature that changed the state of play entirely. The study was led by Peter Hoare at Oxford and Henrik Moritzen at Oldenburg, 2 careers converging on a single experiment that had taken years to design and execute. Jingjing Xu, a postdoctoral researcher working across both laboratories, was first author. The team had accomplished something that the field had been chasing since the year 2000. They had purified cryptochrome 4 from the retinas of European robins and measured its magnetic sensitivity directly. The technical difficulty of this achievement is hard to overstate. Cryptochrome 4 is expressed at low levels in the avian retina. Extracting it, purifying it, and producing it in quantities sufficient for spectroscopic measurement required recombinant expression in insect cell cultures. Growing the protein artificially, then isolating it through a painstaking series of biochemical steps. Once purified, the protein had to be handled in near darkness, because any stray blue light would trigger the very photochemistry the team was trying to study under controlled conditions. When blue light was shone on the purified robin cry 4A, the protein produced radical pairs. That alone was expected. Cryptochrome had long been known to generate radicals upon illumination. But the critical question was whether those radical pairs responded to a magnetic field at Earth field strength. The team used a technique called transient absorption spectroscopy, measuring the population of radical pair states at precise time intervals after the initial light pulse. They applied a static magnetic field of 50 microtesla, the strength of the Earth's field, and compared the radical pair dynamics with and without the field. The results were clear. The applied magnetic field altered the interconversion between singlet and triplet states in robin cry 4A. The effect was measurable, reproducible, and consistent with the theoretical predictions that Hoare himself had been refining for over a decade. For the first time, a protein extracted from a migratory bird had demonstrated magnetic sensitivity at geomagnetic field strength in the laboratory. But the paper went further. As a control, the team also expressed and purified chicken cry 4A, the equivalent protein from a non-migratory species. Under the same conditions, the chicken protein showed no significant magnetic field effect. The robin's cryptochrome was magnetically sensitive. The chicken's was not. The difference appeared to lie in subtle structural variations. Changes in the amino acid residues surrounding the flavin-tryptophan chain that affect the kinetics, the spin relaxation rates, and ultimately the magnetic sensitivity of the radical pair. Xu and colleagues also tested a mutated version of the robin Cry4A in which the terminal tryptophan of the electron transfer chain was replaced with a different amino acid, blocking the final step of the cascade. In this mutant, the magnetic field effect vanished. The result confirmed that the full tryptophan chain is essential, that the compass depends not just on the creation of a radical pair, but on the specific spatial arrangement and electron transfer pathway that cryptochrome 4 provides. The 2021 paper did not prove that Cry4A is the magnetoreceptor in a living robin. It proved that it can be. The protein works. It responds to the right field, at the right strength, through the right mechanism, and the sensitivity disappears when you break the chain or use a species that does not migrate. But there remains a gap between demonstrating magnetic sensitivity in a purified protein in a cuvette and demonstrating it in a functioning cell, in a living retina, in a flying bird. The in vitro result tells you the engine runs. It does not tell you the car drives. Since 2021, the evidence has continued to accumulate. In 2025, a study published in ACS Chemical Biology showed that robin Cry4A associates directly with lipid membranes. Exactly the kind of anchoring the compass model requires if the protein is to maintain its orientation within the ordered disc membranes of photoreceptor cells. The same year, a paper in the Journal of the Royal Society Interface demonstrated that Cry4B, the sister isoform, lacks the photochemical and structural properties needed for magnetic sensitivity, narrowing the candidate list further. And in December 2024, a study in Nature Communications introduced a new theoretical dimension. A team led by Robert Winkler proposed that the quantum Zeno effect, a phenomenon in which frequent measurement or interaction prevents a quantum system from evolving away from its initial state, could play a role in sustaining the coherence of cryptochrome radical pairs far longer than thermal noise alone would permit. If correct, this would help explain one of the most stubborn puzzles in the field: how a quantum process can maintain coherence long enough to function as a compass inside the warm, noisy environment of a living cell. The picture, piece by piece, is becoming remarkably detailed, but it is not yet complete. There is a second magnetic sense inside the bird, and it does not live in the eye. Long before cryptochrome became the focus of the magnetoreception field, a different hypothesis held sway, one built not on quantum physics, but on the most ancient and straightforward of magnetic materials, iron. Magnetite, the mineral iron 2-iron 3 oxide, is the most magnetic naturally occurring substance on Earth. It is found in lodestone, the rock that gave humanity its first compasses thousands of years ago. And in the early 1970s, the microbiologist Richard Blakemore discovered something extraordinary. Certain species of aquatic bacteria contain chains of magnetite nanocrystals inside their cells, aligned like beads on a string, which cause the entire organism to orient passively along magnetic field lines. They were called magnetotactic bacteria, and their discovery opened a door. If bacteria could build magnets, perhaps birds could too. The idea took hold quickly. In the late 1970s and early 1980s, researchers found trace amounts of magnetite in the tissues of a remarkable range of animals: honeybees, salmon, sea turtles, homing pigeons. The deposits were tiny, often just nanometers across, but they were there. In birds, the magnetite was concentrated in the upper beak, embedded within the tissue of a structure innervated by the ophthalmic branch of the trigeminal nerve, a nerve that carries sensory information from the face and beak directly to the brainstem. The anatomy was suggestive. A magnetic mineral in a defined location connected to the brain by a dedicated nerve. It looked like a receptor. But the magnetite hypothesis ran into problems early and has never fully escaped them. The iron deposits in the upper beak are sparse, inconsistent between individuals, and extraordinarily difficult to study without contamination. In 2012, David Keyes and colleagues at the Institute of Molecular Pathology in Vienna published a high-profile paper attempting to locate and characterize the magnetite-containing cells in pigeons. What they found instead were macrophages, immune cells that had simply ingested environmental iron particles. The supposed magnetite receptor cells could not be reliably identified. The result did not kill the magnetite hypothesis outright, but it wounded it severely. More damaging was the evidence from the trigeminal nerve itself. In 2004, Moritzen and colleagues severed the ophthalmic branch of the trigeminal nerve in European robins, the very nerve that connects the upper beak, where magnetite is found, to the brain. If the magnetite system were responsible for the compass, the birds should have lost their ability to orient magnetically. They did not. Their magnetic compass worked perfectly. What changed was something subtler and, in its own way, more revealing. The birds lost the ability to detect small changes in the intensity of the magnetic field, the kind of gradient information that would tell them not which direction they were facing, but where they were relative to the magnetic equator and the poles. This is the distinction that now defines the field's working model. The bird does not have one magnetic sense, it has two. The radical pair compass in the eye, built on quantum coherence in cryptochrome, provides directional information. It tells the bird which way to fly. The magnetite system in the beak, operating through entirely classical physics, provides positional information. A map sense, an awareness of magnetic field intensity and possibly inclination gradient, telling the bird where it is along its migratory route. One is a compass, the other is a map, and they operate through completely different physical principles carried by completely different neural pathways, processed in completely different regions of the brain. This two-system model is now the dominant framework in the field, though it is far from settled. The magnetite map sense remains the less understood of the two, partly because the receptor cells have never been definitively identified, and partly because magnetic intensity gradients are subtle and difficult to manipulate in controlled experiments. Some researchers remain skeptical that the map sense exists in the form currently proposed. A 2025 commentary in the Journal of Experimental Biology, authored by Treiber and Keyes, argued that the magnetite hypothesis has been elevated to the status of a ruling hypothesis, accepted more out of convention than evidence, and called for more rigorous testing of its foundational claims. The criticism is valuable because it keeps the field honest. Science does not progress by consensus alone. It progresses by testing its assumptions, especially the ones it has stopped questioning. The radical pair compass has now been tested at nearly every level, from the physics of spin, to the chemistry of the protein, to the anatomy of the neural pathway, to the behavioral disruption caused by radio frequency noise. The magnetite map sense has not been subjected to the same depth of scrutiny. That is not a failure of the hypothesis, it is an invitation. What remains then is not a single unanswered question, but a constellation of them, each one pulling the field in a slightly different direction, each one revealing how much is still unknown about a sense that has been hiding in plain sight for as long as birds have flown. The deepest of these questions is also the most human. What does the bird actually see? If the radical pair compass works as the models predict, then what it produces is not a number, not a heading, not an arrow pointing north. It produces a pattern, a modulation of the visual field, a symmetric filter overlaid on ordinary sight that changes as the bird turns its head relative to the Earth's magnetic field lines. Theoretical simulations have been built to visualize what this pattern might look like, and the images are haunting. A dark band perhaps running through the center of the visual field along the axis of the field lines, surrounded by a brighter ring that fades toward the periphery. Or a more complex pattern, rings within rings, shifting fluidly with every movement of the head, present at all times, as constant and as natural as the color of the sky. But these are models, not observations. No human has ever experienced this sense. No instrument has recorded what the retina transmits to cluster N when a robin orients on a cloudy October night. The question of subjective experience, what philosophers call qualia, is unanswerable through physics alone. We can trace the photon as it strikes the flavin. We can model the singlet-triplet oscillation. We can map the neural pathway from retina to visual wulst. But somewhere along that chain, the signal becomes experience, and that transition, from quantum event to conscious perception, is a boundary that science has not yet learned to cross. There is a strange poetry in this. Quantum mechanics, the theory that governs the behavior of particles too small to see, operating inside a protein too small to see, inside a cell too small to see, producing a sense that no human has ever experienced, and the whole system works. It works in the rain. It works under cloud cover. It works across 10,000 kilometers of open sky, guiding a bird that weighs less than a handful of coins from Scandinavia to sub-Saharan Africa, and back again, year after year, with a precision that satellite navigation struggles to match. And it has been working for a very long time. The migratory songbirds that carry cryptochrome 4 in their retinas today are the inheritors of a lineage that stretches back tens of millions of years, deep into the Cretaceous, when the ancestors of modern passerines first took to the skies above a world unrecognizable from our own. The radical pair mechanism did not appear suddenly. It was shaped by natural selection over geological time, refined and optimized through countless generations of birds, navigating by a sense so subtle that it took human science 60 years even to suspect it existed, and another 20 to begin understanding how it works. Klaus Schulten did not live to see the 2021 paper that vindicated his proposal. He died in 2016 at the age of 69, still working at the University of Illinois. The idea he put forward in 1978, dismissed by science, ignored for 2 decades, rescued by a student who saw what others had missed, is now the foundation of an entire field. It is taught in universities. It appears in textbooks. It has inspired a generation of researchers working at the boundary between quantum physics and biology, in a discipline that barely existed when Schulten first imagined it. The robin does not know any of this. It does not know about singlet states or tryptophan chains or the lifetime of a radical pair. It knows only that when autumn comes and the nights grow long, something stirs. A restlessness, an ancient pull, and that when it lifts into the darkening sky and turns its head, something in its vision shifts, and the way forward becomes clear. There are those who say that quantum mechanics has no place in biology, that the warm, wet chaos of a living cell is too hostile for quantum coherence to survive, that nature cannot possibly have built a device this delicate, this precise, this dependent on phenomena that physicists struggle to maintain in laboratories cooled to near absolute zero. The robin turning its eye toward the horizon on a cool September evening is the answer to every one of those objections. Not in theory, not in simulation, in feathers and flight and 60 million years of proof. We do not yet fully understand it. The protein has spoken, but it has not finished speaking. The neural pathway is mapped, but its language is not yet translated. The compass works, but its finest details, the exact geometry of the pattern, the precise threshold of sensitivity, the mechanism by which coherence survives the thermal storm of a living cell, remain open questions, each one a doorway into territory that physics and biology have only just begun to explore together. And perhaps that is the most remarkable thing of all, that a creature small enough to hold in the palm of your hand carries within its eye a piece of quantum machinery more sophisticated than anything human engineers have yet built, and that after 60 years of searching, after rejected papers and silent decades and breakthroughs that arrived one hard-won experiment at a time, we are only now beginning to see what the robin has always known.